Thank you, organizers, for the opportunity to present my work. As of April this year, more than 100 million people have been infected with SARS-CoV-2, and about 2 million people have died from SARS-CoV-2 infection. Novel vaccines have been developed and deployed rapidly to counteract the virus. However, strength and durability of antibody responses elicited by vaccines remains uncertain. And so, new research tools are needed to evaluate humoral responses against SARS-CoV-2 infection and vaccines. It has been well established that receptor binding domain and host ACE-2 interaction is critical for entry into target cells. As such, neutralizing antibodies against SARS-CoV-2 that are targeting the spike protein have been hugely associated with protection. More importantly, receptor binding antibodies contribute to the majority of neutralizing activity. And so we want to develop methods to evaluate receptor binding targeting antibodies elicited following SARS-CoV-2 infection and vaccines. And so we developed this Luminex bead-based ELISA to detect Spike-RBD antibodies. So in this assay, we have coupled beads with Spike-RBD. And so in presence of Spike-RBD in plasma from infected patient or vaccinated individual, these antibodies can bind the Spike-RBD, and we can detect and quantify these antibodies using secondary antibodies in this detection Luminex instrument. So we began by validating our coupling approach. Mind you, these are Spike-RBD proteins that are his tagged, so we detected for the his tag in these proteins. You can see that across all these 6 proteins, the signal is significantly higher compared to the background, suggesting that our coupling approach is working really well. And we also went on to use monoclonal antibodies to test whether our assay can perform a dose-dependent antibody detection. So we have the antibody concentration at different dilution or concentrations, and you can see that they correlate very well with the Luminex signal, the fluorescence intensity signal from our Luminex instrument. Please note that similar observation was made when a rapid polyclonal antibody was used. We further went on to test whether our assay can be very specific in terms of discriminating antibody responses against these other human coronaviruses. So we use this human monoclonal antibody. Please note that this antibody can cross-react with SARS. As you can see across these 6, these 2, SARS-CoV-2 and SARS, have very high signal, significantly high signal compared to the rest of the human coronaviruses protein that are close to the background. And so our assay can accurately quantify and discriminate humoral responses among coronaviruses. To test the utility of our assay, we obtained samples from COVID Immunity Task Force study. So in this study, strength and durability of vaccine responses are monitored among BC long-term care residents and healthy care workers. And so please note that our sampling is based on a vaccine dosage schedule in BC. So for instance, visit 1 is a baseline and visit 2 is is a sample you collect for 4 weeks following first dose. So we started by analyzing visit 1 baseline participants, 34 participants, whose visit 2 also samples are also available. So we did this using Roche assay and Luminex assay. Mind you, Roche assay is a clinically validated assay that detects total anti-S receptor binding antibodies, and in this assay, and protein reactivity reflects prior COVID infection. You can see that between these two assays, the data is very consistent. So we went on to also do an analysis for visit 2 sample, that is 4 weeks post-vaccination. You can see that with visit 2 in these two assays, the responses are significantly higher compared to visit 1, and the data is consistent between these two assays, which can be clearly appreciated with this correlation analysis here. Thereafter, we wanted to adapt our assay to be able to determine the level of ACE-2 blocking antibodies that will be predictive of neutralization potential. Basically, we are building on these previous studies, which showed that ACE-2 binding antibodies contribute to majority of neutralization activity in plasma, and that signal decrease in presence of ACE-2 correlates with neutralization potential. And so this is how our assay looks like. So you have a direct signal in absence of ACE-2, and you have a like indirect signal, that the signal you get in presence of ACE-2. So a signal decrease will be the difference between a direct signal and the signal in presence of ACE-2. So again, we validated this assay using commercial antibodies, and also we validated this assay using previously characterized convalescent plasma. So we had 5 neutralizers and 5 non-neutralizer, and these individuals were characterized using a pseudovirus neutralization assay. And you can see that the signal decrease among neutralizers is significantly higher compared to non-neutralizers, suggesting that our assay could indeed predict neutralization potential. And so we went on to test visit 2 samples, and this is 4 weeks post-vaccination again, and you can see that in presence of ACE-2, there's a signal decrease indicating presence of ACE-2 blocking antibodies. And we went on to do a comparison between visit 2 and visit 1, whereby you can see significantly higher amount of signal decrease in visit 2, which is consistent with higher levels of neutralizing abilities as indicated by this pseudo neutralization assay. And so signal decrease would indicate presence of RBD targeting antibodies predicting neutralization potential. So we have established an assay to detect SARS-CoV-2 antibodies. Similar assay can be used to detect presence of antibodies that can compete with SARS-CoV-2 for ACE-2 binding, thus predicting patient serum neutralization potential. And this new research tool should enhance our ability to evaluate the efficacy and durability of current COVID-19 vaccines, and provide a new platform to test future vaccine candidates and antibody-based therapeutics. Thank you.